So next up, we've got our main speaker for today, so Professor Natalie Fay. So Natalie is a Professor of Chemistry in the School of Chemistry at Bristol, with an interest in using data science and computational approaches as a driver for scientific discovery, specifically when applying computational structural chemistry to the design of organometallic catalysts. So over to you, Natalie.
Thanks very much, Adam. And let me start out by thanking the team for organising this webinar. I've had a look through the list of people, and it's always nice to see a lot of familiar names. So as you can see from the title, I was thinking of the sort of hybridization of plants and looking at botany a little bit when I came up with this title. So this idea of saying, okay, what happens when we cross organometallic chemistry with data science approaches. I promise I'm a whole lot less machine learning than my predecessor today. But one of the things that started to cross our minds was this distinction. So when I started out using data-led approaches in combination with chemistry, it was very much a case of saying, okay, we'll just do some statistical analysis. But the field has most certainly moved on. And we've come from an area of just talking about statistics, which you might broadly define as drawing conclusions about global populations from small samples, to actually talking about data science as a field. And that's much more drawing on growing computing power, but also software developments and visualization. A lot of the stuff we're doing is actually about getting a handle and visualizing chemical space. For those of you who are interested, one of the first people who looked at data visualization and this broader idea of data science was actually Florence Nightingale. And she was interested in convincing people that infection prevention and better hospital hygiene was actually almost more important than treating the immediate injuries of soldiers that had been evacuated from battlefields. So I've put a couple of links in there, and I can share them later, or maybe Azel can send them around. They are just some relatively recent articles about that aspect of data visualization. And it was John Tukey who really coined the term data science. So from our perspective, one of the big areas we've been working in Bristol, and that's been going on for quite a long time, is this idea that molecules constitute a chemical space, and that perhaps you can use structural and electronic data to actually map out this type of chemical space. And I can illustrate that a little bit more. So our main area of interest is organometallic chemistry. So when I say ligand, I mean small molecules that coordinate to a transition metal center. And in organometallic chemistry, those ligands are often used to fine-tune the complex properties. So you would, for example, have a metal complex that is catalytically active, but to try and improve that catalytic activity, it's quite a common strategy to swap out the ligands and thereby, for example, to make them more electron rich, more electron withdrawing, or to modify the size of the coordination sphere around the metal center. And this isn't a new idea. So this goes back to at least the 1970s. You can characterize these ligands in terms of both their size and their electronic properties. So what Chadwick Tolman did quite a long time ago in this ChemRef was to actually formalize measures for the size of the ligand. So you've got a steric measure, generally called the cone angle, where you tuck in all of the substituents on your phosphorus donor atom. You place the phosphorus donor at a fixed distance to a nickel center, and then you measure the outside of the cone that is formed by the most outlying atoms on your ligand. And that's a fairly decent measure of the size of your ligand. It has some drawbacks, but it's very well established. We also have something that's nowadays called the Tolman electronic parameter. and that's the highest carbonyl stretch in a nickel tricarbonyl phosphorus donor ligand complex. And it's basically a measure of the net electronic effects. And again, there are perhaps some drawbacks to that measure, but what you end up with then is a steric dimension measured by the cone angle and an electronic dimension measured by what is now called the Tolman electronic parameter. If you plot them against each other, then you have what you might call a map of that phosphorus donor ligand chemical space. And you might notice a few things about this. So ligands that from a chemical perspective, we might describe as similar, cluster quite close together. You've got your alkyl phosphines down here, ligands and phosphides up here. Ligands that are quite different from each other, for example, PF3 is an outlier here, are very, very far apart. The other thing you might notice is that there are big areas of this chemical space where there are no ligands detected in these parameters. So there may well be areas of that chemical space which are not fully sampled. And that's something we can come back to later on. Now, you've characterized your ligands in terms of sterics and electronics, and you can then also try to derive structure property relationships. So saying, for example, if I make my ligand 10 times bigger, what will be the effect on a rate of a reaction or some other measurable experimental quantity? And that's really what makes these processes quite interesting. So in our own work in Bristol, we've built a number of databases, and it's not my fault, but they're called ligand knowledge bases, abbreviated to LKB. And I'm going to walk you through our bioidentate database quite quickly, so we can then talk a little bit more about some newer work we've done in this area. So the idea is that you calculate the same ligands, both as free ligands, so here free to move, and here in the coordinating geometry, the ligand coordinated to representative metal fragments, in this case, a zinc dichloride complex, which imposes no crystal field stabilization, and a palladium dichloride complex, which is square planar and has quite a strong crystal field stabilization energy. We have done some work on splitting ligands in half and having descriptors for each of the donor atoms. So that's what this truncated ligand part is about. And then we also have developed a couple of ways of looking at the steric size of that ligand, both in a rigid way, which is the helium-8 wedge, and then in a way that it's free to adjust to steric pressure in the coordination geometry, which is labeled N helium-8, and that's just the new helium-8 parameter for those of you who might be interested. We run very, very standard DFT, and it's a big shift from the world of people who argue about density functionals towards saying we just need a computational approach which is suitable and which doesn't create any unusual behavior. So when we started that work, which is longer ago than I care to remember, BP86 was quite a good shout because it was robust and it wouldn't do anything crazy. So you run these calculations and you extract data from those calculations, and that will be things such as the binding energy to the different fragments, the interaction energy with those helium-8 wedges, and a range of structural descriptors that essentially capture the change of both the ligand and the metal fragment on coordination. And this initial work was predominantly for phosphorus donor ligands, but we threw in some phosphorus nitrogen donor ligands as well. The 28 descriptors are sometimes a little bit challenging to visualize. So to deal with this, we used one of the OG projection methods, in this case, principal component analysis. And the idea is that you reduce the number of dimensions by transforming your original data, and the goal is to optimally represent the distances between objects. So for principal component analysis, you actually work with linear combinations of the original variables, and that will become later on. The goal is to place most of the variance in the data set into the first principal component and the next highest amount into principal component 2, and so on and so forth. So decreasing importance of those principal components. And the other bit is that they're designed to be orthogonal to each other. So you might be used to looking for linear relationships and regressions, but in this particular case, we actually want a plot that is very well filled rather than having some trends in the data. So if you perform this type of analysis on our original database, and I'm just showing you the data for the phosphorus-phosphorus donors alone, so there are no phosphorus-nitrogen ligands in there, then you find that the first two principal components cover about 58% of the variation in the data set. So that's reasonably good for reducing from 28 dimensions down to, in this particular case, just 2. The spots, each spot corresponds to data for one ligand processed with principal component analysis, and they're color-coded by the substituents on the phosphorus atoms. So you can see, for example, here the black squares are alkyl phosphines, the blue dots are aryl phosphines. We've got phosphides over here. Phosphorus in an aromatic ring is sometimes quite interesting. The other thing you might notice is that, again, there are large areas and smaller areas as well of this chemical space which don't have any known ligands in them. So that phosphorus-phosphorus space is perhaps not sampled very well. One of the things we did quite some time ago now, it was published in 2013, was I worked with a postdoc working with me at the time, Jesus Robert, on basically looking at, well, what could we do to improve that sampling of the chemical space? And we chose to treat ligands like Lego bricks. So you've got your little cartoon up here, changing the backbone. We kept the donor atoms the same, but we also changed the substituents with the constraint that the substituents on both donor atoms would stay the same. So we ran these calculations, and it was 275 ligands that we looked at, and processed the resulting data, which was, it's the same descriptors, the same approach as I showed you for the more chemically driven phosphorus donor database. But we took out the data, we processed it with principal component analysis, and this is the resulting chemical space. And in this particular case, it's color-coded by the 11 substituents we looked at. So you can see we've got some alkyl substituents, we've got some more electron withdrawing groups on here, and a range of aryl groups on there as well, both standard phenyl and then perfluorinated, and then some ortho and paratolar substituted ones. So if you look at this across the chemical space, then you can see that the principal component analysis is detecting that at least some of the variation in this chemical data set comes from those substituents, and you see these bands in chemical space. So it's picking apart those chemical groupings, similar to what I showed you on the Tolman maps before. The other dimension of variation was the ligand backbone for us, and we sampled in total 25 different backbones, and they are shown over here. And I can promise you, if we color-coded this particular plot by backbone, it becomes unintelligible. So what we did instead was we color-coded by the length of the backbone. And you can again see that you see these bands across the chemical space, this time going across the other diagonal. So substituents effects go in this way. The length of the backbone to some extent goes in this way. If you're particularly eagle-eyed, you might see that especially the really long 8-membered backbones show up almost everywhere in this chemical space. And that's because they're actually quite flexible. And the new helium-8 parameter allows us to take that flexibility into account. So they will bend themselves to the available steric space in that ligand, and thereby be more dominated by the substituent effects. So one of the things we concluded from this was that whilst a lot of synthetic chemists focus on varying the backbones, you could actually increase the variability across that chemical space by also playing around with substituents. And one of the drawbacks of doing something like this in a very computer-led way is that, of course, not all of those backbones, and the backbones themselves are synthetically accessible, but it's not necessarily straightforward and easy to actually make those ligands and to measure their properties. So there is no control for synthetic accessibility in this particular database. And I'm well familiar with examples where synthetic chemists are quite convinced they can make something, and then 2 years later they will come back and say, no, actually that's not going to happen after all. So as time has gone on, and especially accelerated by COVID, lots of other groups have started to look at data-led approaches. And one of the things people started asking me was, have we tried other dimensionality reduction approaches beyond principal component analysis, which has been around for a very long time, is quite well understood, but to a lot of people also feels a little bit old-fashioned, perhaps. So we had a very talented student visiting us from Spain, Mario Villares-Canon, and Mario was interested in using different dimensionality reduction approaches on ligand databases. So my postdoc at the time, Carla Saunders, and I decided that we could have a little look at some other dimensionality reduction approaches beyond principal component analysis. So principal component analysis is a linear approach that makes it easier to interpret in some settings. But two of the more fashionable and newer approaches that people started to use, including in the context of organometallic chemistry, were TSNE, so T-distributed stochastic neighbor embedding, which is a local but non-linear approach, and then also uniform manifold approximation and projection, UMAP. And that captures both global and local structures, and again, is non-linear. So we use those two approaches on the same data sets. So the color coding is the same, the data are the same, they're just processed with different approaches. So this map, which is the principal component analysis, should look familiar to you. For ease, I'm also showing you the first two dimensions in UMAP and in TSNE. So down here, you can see UMAP, and UMAP is good at clustering. And I think you can see that here quite strongly. So especially the electron withdrawing substituents that the principal component analysis places on the edge of the chemical space become even more tightly clustered and to some extent also more isolated if you run UMAP on them. TSNE for us didn't look like an improvement. You don't get tighter and clearer clustering than you do in principal component analysis, and you don't get that interpretability in quite the same way. So we did look at some TSNE and we smoked, but we didn't inhale in this particular case. But we did do extra work with UMAP. So if I just bring those two approaches up in a slightly bigger way, what you can hopefully see and agree with me is that in both cases, the different substituent types get clustered quite well. And that shows us that our underlying calculated DFT descriptors are probably capturing the key aspects of the chemistry, making it relatively straightforward for these approaches to detect the differences. And, you know, especially for principal component analysis, I showed you the effect of different substituents and backbone length, but you can, of course, then also relate that to the bite angle, the size, and the electron withdrawing properties of your ligand. So although we're using different techniques, ultimately, we're actually seeing quite similar clusters with the UMAP approach clustering some of the more exotic or more unusual ligand types a little bit more tightly. The other thing we often do is we color code our maps of chemical space by individual parameters. So the first color coding here is by that new helium-8 parameter. So it measures the size of the ligand, essentially felt by the interactions between the ligand and the other ligands coordinated to the metal in cis positions. And you can see that there's this trend across that chemical space with principal component analysis, with very large ligands being near this end and the smaller ligands being down here. UMAP shows clustering according to size as well, but remember it's a non-linear technique, and in this particular case it's much more dominated by these tighter clusters of electron withdrawing ligands. So the next parameter we color coded by was the proton affinity. And that's the proton affinity of one of the donor atoms, but it correlates with the other donor atom as well. Most of the ligands, well, all of the ligands in this one are actually symmetrical. So what you can see is that the high proton affinities are clustered in this area of the principal component chemical space, whereas they're up here. And then again, these outlier clusters of ligands have more electron withdrawing ligands more electron withdrawing substituents on there. The third parameter we wanted to explore was the bite angle on zinc. So that's quite close, but not identical to the natural bite angle of ligands. There's no crystal field stabilization energy, so the bite angle can go as big as it needs to. And it is both capturing steric and electronic effects. And again, you can see that there is grouping across that chemical space, and the same thing really happens with UMAP, but it is perhaps not as drawn out as other things. So that's looking at things from a data-led perspective, but ultimately our philosophy has always been to try and do useful things in terms of chemistry. So we also had a look at some common ligand variation. So ligand 1, for example, is this short backbone, a single carbon atom, and the phosphoruses are methyl substituted. So you can then explore the effect of what happens if you go to a longer backbone. So we go from 1 to 12, and the same thing you can observe on your UMAP map. You can look at what happens if you go from methyl substitution to phenyl and to tertiary butyl. So you're moving across that chemical space, changing both the size and the electronic properties of the substituents. You can look at backbone K and compare tertiary butyl, that's 112, with phenyl substitution, that would be 117. So again, you're seeing that change in electronic and steric parameters. And then you can also look at that much more rigid backbone and find that actually there are some similarities that are coming about in that chemical space, even though that might not be entirely intuitive. So we thought that was quite an interesting one because remember when I first talked about mapping this ligand space and ligand properties, I mentioned this tunability, this idea that you can change the substituents, you can change your ligand, and you can fine tune your catalyst properties. And essentially that's what we're seeing in this chemical space here. That for some areas, and it doesn't matter how you map it, but for some areas of this bisphosphine chemical space, there are lots and lots of ligands of very similar properties. So chemically, that would give you the impression of nearly endless tunability. And then there are some other areas of the chemical space, and especially the more electron withdrawing substituents, where you're actually seeing less continuity. There are big gaps in that chemical space, and it is entirely possible that other substituents and other ligands will fill those gaps, but it isn't as immediately obvious how you tune your way from one property to another when there are those bigger gaps. So I thought that was, from a chemical perspective, a really interesting outcome, and certainly potentially of use to people looking to optimize their catalysts as well. We also looked at wider bidentate ligand space. So not just varying the substituents and the backbones, but also playing around with what happens with donor atoms. And you can see here that for some types of donor atoms, and that would be nitrogen, 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 oxygen, and oxygen, oxygen types of donors, they stay quite similar to each other, whereas other completely chemically different donors actually sit further out in that chemical space. And just for completeness, so this is color-coded by donors, this is color-coded by the length of the backbone. And again, you can just about see those bands going across that chemical space. And that really just is principal component analysis doing what it was designed to do, which is detecting what the main sources of variation are. And in this case, the strongest source of variation is the change in donor atoms. So if you do your work carefully, you can actually extend this concept of mapping chemical space to comparing multiple donor atoms as well. And we had these maps, and then at some stage, my colleague Robin Bedford bounced into my office and said, well, you've got zinc and palladium in there to calculate your descriptors, and we can do things with gold atoms as well. I'm interested in first row metals. Indeed, so am I, actually. But Robin's interest is predominantly in first row metals. And what he wanted to know was whether this approach would work for first row metals with no further work, or whether we would need to do extra work, use additional descriptors that can deal specifically with first row metals. And we've recently started a prosperity partnership funded by EPSRC and working with AstraZeneca and Labman to develop first row transition metal catalysis for the pharmaceutical industry. So it turned out to have been a really, really timely question. Before we knew that the funding bid was successful, I went to one of the ACS meetings and I got chatting to Nile Hazari and Liv Miritza, and they both came to me and they said, this is all very nice, but will it work for first row transition metals? And specifically, Nile had done some work looking at some BP type ligands with different substituents and observing at what point of modulating the steric and electronic effects you would actually see a switch from high spin nickel tetrahedral complexes to low spin nickel square planar complexes. Liv, on the other hand, had designed this pyridinophane type ligand, and I'll show you what the structure is in a second. Should have put it on this slide. But Liv had designed this particular type of ligand specifically to support low spin square planar coordination. So they're both interested in cross-coupling types of reactions and controlling them through choosing your nickel catalyst particularly well. So I had a project student last year, Sarah, who worked her way through all of these different nitrogen donor classes. This, by the way, is Liv's strapped pyridinophane type ligand, and add them to our bidentate multi-donor database. And if you recall, we had a fairly balanced database with some nitrogen donors, and Sarah's work put in quite a lot more of these nitrogen donor ligands. And we can spin this out and just look at those, but we actually want to go to the wider chemical space in due course. So most of those blue spots are Sarah's work. And what we were interested in was whether we could correlate the original ligand knowledge base data with the energy difference between a tetrahedral nickel-2 complex. So that would be the product of the oxidative addition of an aryl halide versus a square planar nickel-2 complex. And basically, it's color-coded again. And the redder colors favor low spin configurations, whereas the greeny and yellowy colors are heading for the for high spin. So these ligands down here, I think I've got the structure with me this time. I have the structure in my back pocket. So these guys down here are Liv's ligands. So these weird ferrocinophanes. What we were hoping for was a clear trend across that chemical space, perhaps leading just in one direction to distinguish between high and low spin. And I think you'll probably agree with me that there is some clustering of ligands that are closer to preferring low spin configurations, but there is quite a lot of noise and quite a lot of variation. One of the things we did again, and I warned you about this, was to look at the relationship between this particular spin state energy difference and some individual parameters. So this is our helium-8, our new helium-8, the flexible parameter. And you can see that the pyridinophanes are weird because they're all really, really rigid in structure and basically all have the same size. So there's very little variation with substituents. But for the other types of ligands, you're seeing some form of trend across the chemical space. It's noisy, it's scattered, but it actually ties in with the work Nile has done in terms of varying the substituents and looking at the impact of that variation on the spin state. So we are seeing a relationship with steric effects. We also looked at the binding energy on this palladium parameter, and again, you see some variation, and this time Liv's ligands are almost behaving themselves. So you see some response of that spin state energy difference to the electronic properties as well, and then net electronic properties in this case. So one of the other questions that came up in this context was to argue whether the unpaired electrons actually solely sit on the metal, or whether these might be complexes where there's ligand non-innocence. So some unpaired electron density on the ligand rather than on the metal, meaning the formal spin state of the metal might not be what we're expecting it to be, so nickel 2, but instead you've got some unpaired electron density on the metal. And we had a bit of a rummage, and John McCready and Jose Goya-Cochea looked at this, the relationship between neutral bipyridine type ligands and radical anions in this case. And I thought that might make a cute descriptor. And we then had a look for these different classes of ligands, whether changing the substituents to be either really good electron donors or very, very electron withdrawing would give us a noticeable change in the energy difference between the neutral versus the radical anion species of the ligand. And that's nothing to do with the metal. These are calculations just on the ligand. And this is work Felix McFall helped with over the summer, funded by an RSC bursary. And as you can see, there is clearly a trend across there. So that's work we are now continuing to develop, including as part of the Prosperity Partnership. We have done other databases. This is an example of our monodentate phosphorus donor map. And you can see we used related but slightly different complexes in this case. And this is by far our most well-developed map. And we've done work there to see whether you can map experimental data onto this map, in this case for palladium catalyzed cross-coupling reactions, and then also whether you can train different types of models. In this case, I'm just showing you a linear regression model to predict the behavior of catalysts, including the ligands, across the rest of the chemical space. And it's not the greatest model in the world, I won't lie, but it actually gives you some idea about the behavior across the chemical space. Similarly, you can relate calculated data to this chemical space. So in this particular case, we looked at the preference between monoligated oxidative addition, and in this particular case, bisligated oxidative addition, and it is simplified. There's a third possible pathway. But we chose ligands to calculate with standard DFT across all of the chemical space. And basically the ones marked in red favor the low ligation states, and chemically they're large and electron rich. And the blue ones actually favor the bisligated pathway. This is a projection of experimental high throughput experimentation data. And you can see again that we've got this hotspot down here of large and electron rich ligands. And you can train a different type of predictive model on this calculated data at the top, and again get a prediction across this chemical space that helps us relate this to the mechanistic finesses of cross-coupling reactions as well. So it is quite well known that good catalysts tend to favor a lower coordination number on your palladium. One of the things we've been interested for a long while was to improve the mapping of the commercially available chemical space. And my student Nick, who had his PhD viva on Monday and has some work to do, actually looked at this. So this is the map I've already showed you, give or take a few ligands, and only 94 of those ligands are actually commercially available. And in some areas of that chemical space, there are big gaps. We've done quite a lot of work to beef that up. So this is one version of the updated map, and it's showing 809 ligands, 223 of those are commercially available. One thing that might interest you is that there are still areas of chemical space where it's not much commercially available. But this area of large electron-rich ligands, important for cross-coupling reactions, is actually quite well populated in this case. And I'm just in the process of writing this up to have another update on our monodentate map. And annoyingly, I've had to do some data cleaning, and it's not a beautiful, here is an update taking us to 800 ligands at the moment, but 796. I'm really, really cross about that. I might go and find 4 more ligands to add as well. So very quickly, I'm conscious that there's not much time left. We are also looking at using the computational mapping approaches to learn more about reactivity. So all of the ligand maps are about ground states or about making predictions based on ligands for reactivity. But we've also looked at actually calculating barriers and seeing whether we can use this to map the chemical space. So in this particular project, we have looked at cationic rhodium complexes, and the idea was to computationally find applications for known catalysts that might do something interesting. So this ran through the pandemic with my former student, Derek, and he calculated barriers for all of this postulated catalytic cycle, looking at different variations. So whether the solvent is coordinated first or whether the alkene is coordinated first, and then working through all of these catalytic cycles. And I'll just quickly show you some of his results for oxidative addition. So we looked at the activation of all of these bonds shown here to these multiply bonded fragments, as well as in the presence of solvent. And what you can see is that there are some clear trends across the different bond types. And what you find is that actually that correlates quite well with the strength of the bond we are seeking to activate. The other thing you should notice is that I brought in this line, and that's related to what's catalytically actually feasible. So if you want reasonable reaction conditions, so up to about 80 or 90, maybe 100 degrees Celsius, then the calculated Gibbs energy of activation shouldn't really be higher than 30 kcal per mole. So you can see that quite a lot of those substrates are actually ruled out computationally, because the barrier to that first crucial oxidative addition step is higher than what you could reach. So especially for electron-rich substrates, it's not necessarily going to undergo. We did similar steps for the migratory insertion and for the reductive elimination. But eventually, all of that work predicted that hydrogenation and hydrosilylation should go quite well for these rhodium cationic complexes. And when restrictions from the pandemic were lifted, Derek was actually able to get into the lab and try out some of these predictions. And all you need to take away from that plot really is that it's less clean than you would see if it was just a successful rhodium catalyzed silylation. So we found that we had a beautiful house of cards from the computational data, but it didn't really match the experimental data. Going back to the literature, what you then find is that you can get the alkyl silane, but under certain reaction conditions, you can also see alkene isomerization and vinyl silane formations. So it very much depends on finer details of those reaction conditions, which pathway you go around. And then, of course, you can come in, and this was a project student a couple of years ago, and actually introduce additional reaction pathways to your calculations, and that's work we're seeking to continue. Right, so I wanted to talk about what we can do when we cross organometallic chemistry, especially in homogeneous catalysis with data science. And one of the things we're working towards is that idea of finding the best synthetic route, helping chemists to use better resources, but also sometimes to break that cultural attachment of, I've always done the reaction in this way, I will continue to do so. Much more powerfully from an industrial perspective, actually advising people on when they should stop can be quite useful as well. From an academic perspective, we like to broaden our toolkit and to explore and discover new chemistry, but also to build this on deeper mechanistic understanding. And what we're doing is to work with experimental groups to actually work towards reliable computer-based prediction as well. And I give you the little GIF of the spherical cow. So I would say we're probably still on the spherical side of the cow rather than having a really good model and a description of all of the chemistry, but we're definitely working towards that in the right direction. So I don't do most of the work myself anymore. All of these folks have contributed significantly to the things I've showed you today. I have a number of fruitful collaborations with colleagues in Bristol, both past and present, but also lots and lots of really good experimental collaborators. And some of the work I showed you goes back to my fellowship, where we worked with this team from AstraZeneca. We're based in computational chemistry, and we've had money from lots of people. And I've very briefly flashed this up. We are promoting flash communications for organometallics, but with this, thank you very much for listening.